Ever wonder how scientists find those, well, those super microbes? The ones that can, like, shrug off poisons or live in really weird places?
Yeah, it's like finding a needle in a haystack, but instead of needles, it's a microbe with some really cool new ability.
Exactly. And today we're going deep, deep into the world of mutant microbes. Specifically, how scientists screen and select the ones that can basically ignore things that would normally stop them in their tracks, like inhibitors and repressors.
We've got some really cool lab techniques laid out in front of us, so ready to unpack how they find these little survivors.
Absolutely. Let's start with the tough guys. The mutants that just don't care about inhibitors, the substances that usually mess up a microbe's growth or its important jobs.
Right. And one way to find those tough guys is, well, it's called growth selection in inhibitor-containing medium.
Okay, that sounds a bit intense.
It's pretty straightforward, actually. It's like survival of the fittest, microbe addition. You basically create this environment where only the resistant ones can make it.
So you take your microbes.
And you might want to shake up their DNA a little bit first, create some variety with some mutagenesis.
Gotcha. Mix things up.
Yep. And then you put them on a special dish, with the inhibitor added, just enough to wipe out any normal non-resistant microbes.
So it's like a microbial obstacle course.
Exactly. Only the mutants that have figured out a way to beat the inhibitor, they're the ones that will grow and form those little colonies you see.
I see, I see. Then the scientists, they can pick those survivors, double check that they are truly resistant, and then comes the fun part.
Oh, yeah, the real detective work begins.
Figuring out how they did it. Like, did they change the target that the inhibitor was supposed to attack?
Mm-hmm.
Or maybe they developed some kind of a pump, like a tiny little pump to just get rid of the inhibitor.
You got it. Understanding the how, that's the key to figuring out if we can, potentially use that resistance.
That makes sense. Now, I read about another technique for finding those inhibitor-insensitive mutants. And this one, it focuses on a specific enzyme. It's called enzyme activity assay-based screening, if I'm saying that right.
You got it. So instead of just seeing who can survive the poison, you're basically checking if a particular machine, the enzyme, still works, even when the poison is around.
Oh, that's smart. So if you know the inhibitor blocks the specific enzyme.
You can take extracts from your mutant microbes and you test them directly. You see if that enzyme is still doing its thing, even with the inhibitor present. And if it is, boom, you've got a potential inhibitor-insensitive mutant.
Nice. And then I guess the next step is to, like, zoom in on its DNA and figure out what little tweak in its genes let that enzyme keep on trucking.
Exactly, exactly.
All right, so we've talked about those inhibitor-ignoring microbes.
Yeah.
But what about repressors? Are those, like, finding mutants that can do stuff they're not supposed to?
That's a good way to put it. Repressors are like off switches for genes. So finding repressor insensitive mutants means finding cells where those switches are kind of, well, broken.
Interesting. So how do they find those?
One way is through growth selection in the presence of repressing conditions. And a really good example is something called catabolite repression resistance.
Catabolite repression resistance.
Right.
Okay, you're going to have to break that one down for me.
All right, imagine a microbe, and it's got two favorite foods. Let's call them sugar A and sugar B. But it really, really loves sugar A, so much so that if sugar A is around, it won't even bother with sugar B.
Picky eater.
Totally. And that's because of something called catabolite repression. Sugar A basically represses the genes the microbe needs to use sugar B.
So sugar A is like, I'm the favorite. No need for sugar B.
Yeah, exactly. So to find mutants that can resist this, you got to be clever. You feed them a mix of both sugars.
And see if any of them can resist the temptation of sugar A and still munch on sugar B.
Exactly. You're looking for the rebels, the ones that can use both sugars even when sugar A is plentiful. Then you'd pick out the ones showing they're using B even with A around and figure out what went wrong with their off switch for using sugar B.
Cool. So maybe the switch itself is broken, or maybe they can't even sense sugar A anymore?
Right on. Now, another way to do this is even more high-tech. It's called reporter gene assay-based screening.
Okay, now that sounds complicated.
Think of it this way. Imagine putting a tiny little light bulb inside the microbe. And this light bulb, it's connected to a gene that's normally turned off.
A light bulb inside a microbe.
It's not a literal light bulb, but you get the idea. It's a signal. So scientists engineer microbes to have this special gene called a reporter gene, and it's linked to the gene they want to study, the one that's normally repressed.
Okay, I think I'm following. So if that normally off gene suddenly turns on, because of mutation.
Then the reporter gene turns on too, and you get a signal. That could be a color change, or sometimes it even glows. This makes it so much easier to spot the mutants you're after.
That is pretty brilliant. So whether it's an obstacle course or a sugary buffet, these methods are all about finding those special microbes hiding in a massive crowd.
Absolutely. And these methods are crucial for all sorts of things, like developing microbes that can resist the harsh chemicals used in industrial processes, or even engineering microbes to produce those important medications under all sorts of different conditions.
It's amazing to think about, really. I mean, if scientists can create this kind of selection pressure in the lab and find these resistant microbes, what does that tell us about how life adapts and evolves in the wild? It's like fast-forwarding evolution right in front of us.
It really makes you think, doesn't it?
It sure does. And that brings us to the end of our deep dive for today. Thanks for listening.
Thanks for having me.